There are different mechanisms through which genetic material can be transferred from one bacterium to another. The first mechanism is transformation, where the naked DNA is picked up from the cell lysis. So since the naked DNA is in the outside environment, if you add deoxyribonuclease to the cell culture, it will degrade the DNA and thus transformation would be stopped. So here is an image that shows transformation. So as you can see, the naked DNA is picked up by another cell and then it's incorporated into the genome. The next mechanism is transduction, where a virus will transfer genetic material from one bacterium to another. And here is an example of transduction. So there are two forms of transduction. One is called generalized transduction. So in the lytic phase, when the virus infects the bacterium and new viruses are formed, some of the bacterial DNA may be incorporated in one of the viruses. And when this virus infects a new bacterium, this DNA could be incorporated into those cells. The other form of transduction is called specialized transduction, which includes the lysogenic phase. So when the viral DNA is incorporated into the bacterial DNA, it can undergo multiple divisions. And then eventually, when the viral DNA is excised out, it may cut part of the bacterial DNA. So the flanking DNA may be cut with it and transfers to the new bacterium. So this is referred to as specialized transduction. Now, there are certain bacteria that produce toxin through lysogeny or specialized transduction, and you can use the mnemonic A, B, C, D, E to remember these bacteria. So A goes for Shiga toxin produced by enterohemorrhagic E. coli, B for botulism, C for cholera toxin, D for diphtheria toxin, and E for erythrogenic toxin of group A, streptococcus. I would also like to mention that the terminology transduction is also used in human gene therapy. So the first gene therapy was done in patients with SCID, also known as severe combined immunodeficiency. And what happened is that in these patients, the bone marrow is extracted, and then the CD34 positive cells are infected with a retrovirus that contain the gene that is defective in these patients. So therefore, the gene would be incorporated into the bone marrow, and these bone marrow are then transfused back to the patient. So therefore, bone marrow are transduced. So transduction is the other terminology that is used in human gene therapy. The next mode of gene transfer is called conjugation, and it's via direct cell-to-cell contact via a sex pilus. So here is an example of conjugation. As you can see, there is a sex pilus that allows the gene to be transferred from one cell to another. And finally, the last condition is transposition. So there are certain DNA segments called transposable elements which contain inverted repeats at the end. And these transposable elements can be jumped from one region of the genome to another, so they keep jumping around. And when they jump around, they may take part of the DNA in the genome with them, and thus they will transfer part of DNA from one region to another or from one cell to another cell. And that concludes our discussion.